Hello everyone, welcome back to the class. So from today we'll be learning a new module, that is cell cycle. So the cell cycle chapter will be dealt in two parts, that is part 1 and part 2. Part 1 is nothing but the basic aspect of the cell cycle and part 2 is regulation of cell cycle. So what is cell cycle? So in this image you can see a single cell is processed to form a two daughter cells, right? Basically cells, or if we have to split the meaning of this word cell cycle, cycle is a sequence of events. Cell is a structural and functional unit of organism. So cell cycle is nothing but the sequence of event of the structural and functional unit of cell. So what happens, structural and functional unit of the organism. So what happens in this sequence of events? In the sequence of events, so the cell duplicates or divides to form two daughter cells. What about its genome? Genome, here present in pink color, it is duplicated and it is equally distributed to daughter cells. So we can say cell cycle is an ordered series of events, or it is a sequence of events by which cell duplicates its genome and eventually divides into two daughter phases or two daughter cells. Okay, there are two phases. There are two phases of a cell cycle. Two phases of cell cycle. Which are they? They are interphase and M phase. For now, M will be defined as mitosis. Okay, so there are two phase, interphase and M phase. So what is this M interphase? Interphase is a phase, or it is a known to be a resting phase. It is a time during which the cell prepares itself for cell division. So cell prepares for cell division, and there are two processes undergoing in this phase, that is cell growth and DNA replication. So there are these two processes happening in the interphase. It is also known to be resting phase. Now what about M phase? M phase is a short phase. It is a short phase where actual cell division happens. Okay, it corresponds to visible mitosis for now. So there is another process or another type of cell division which will be dealt later. So this phase starts with the nuclear division. So what happens here before nuclear division? In that, DNA replication and cell growth was predominant. Here nuclear division, and it corresponds to daughter cells. That nuclear division is known to be karyokinesis, and usually end up this cell division is known as cytokinesis. So M phase is the short phase when it is compared to interphase, where the actual cell division happens. Visible mitosis can be seen. Nuclear division is initiated, or this phase starts by nuclear division, that is karyokinesis, and ends up with cell division, that is division of cytoplasm, cytokinesis. We can modify this cell as cytoplasm, where the cytoplasmic division is known to be cytokinesis. See here, cell cycle is a ordered series of events. It is a sequence of events by which a cell duplicates its genome and eventually divides into two daughter cells. It has two main phases: interphase, where the time during which each cell is preparing for division by undergoing both cell growth and DNA duplication. M phase, the period of actual division corresponding to the visible mitosis, the phase which starts with the nuclear division corresponding to the formation of daughter nuclei, karyokinesis, and usually ends with division of cytoplasm, that is cytokinesis. Here you can see a single cell where cell grows and chromosome replication happens, so we can infer that it is an interphase, and the chromosome is segregated. From here, from M phase it is happening here, that is cell division, proper cell division. The single cell is dividing into two daughter cells. So this is a hypothetical division of eukaryotic cell. So next, now under interphase there are many subphases. Under interphase is a long resting phase where there is two events happening: DNA replication and cell growth. Okay, in interphase, so it is a very active phase. In interphase there are three subphases. They are G1 phase, S phase, G2 phase. Okay, G1 phase, S phase, G2 phase. G1 phase, the G refers to gap here. G1 means gap, S means synthesis phase, G2 means gap 2 phase. So there are three sub-phases in interphase: G1 phase, S phase and G2 phase. Let's discuss this one by one. In G1 phase, what all happens? So it is a period between end of M phase and start of DNA replication. It is a period between. So if this is a M phase and this is DNA replication phase, DNA replication or synthesis of DNA phase, so the period between this is known to be G1 phase. Okay, next is S phase. S phase is the phase where DNA synthesis is happening. Where DNA is synthesized with the help of some components. Okay, then next phase is G2 phase. G2 phase is the gap between following DNA replication and preceding initial M phase. So it is the period from the start of DNA replication to initial M phase. So the phase between DNA replication and M phase is known to be G2 phase. G2 phase. If I have to represent this in a cyclic manner, we can say I'll write the major events happening. So first I'll write M phase, I'll write DNA replication phase, okay. Now I'll connect all these. So the period from M phase, the period between M phase and DNA replication phase is I'll write it in a different color, G1 phase. The period of DNA synthesis or DNA replication is known to be what? So this is S phase. This is S phase. Okay. Then the period, just a minute, I'll write it back. M phase, DNA replication phase. So the period between M phase and DNA replication phase is known as G1 phase, and the period of DNA replication or a period where DNA synthesis is taking place, that phase is known as S phase, and the period between DNA replication and M phase is known as G2 phase. G2 phase. There is another phase that is G0 phase. G0 phase. In this phase there is no cell division. No cell division happening. Then what is it? So it is a arrested state. So there are two types of or two states of G0 phase, that is reversible and irreversible. Reversible is also known as quiescent stage, and irreversible is known as senescent stage. In reversible stage, what is happening? In reversible stage, the cell remains active. They are active, but no longer proliferate. So they don't proliferate. They are not proliferating. At a given time, at the requirement, as per the requirement of organism, it reverses or it starts dividing. So it is a temporary arrested. And irreversible means, it is also known as senescent. Senescent stage. In this stage, cell has ceased proliferation. It is metabolically inactive. Where it is permanently withdrawn from the cell cycle. So it is no longer proliferating, and also it has lost the proliferative potential. And there is another state of G0 phase, that is terminally terminally differentiated state. It is just similar to irreversible, but often differs from it in a small aspect. So here, in this course, cell in this course is acquiring specialized function. So cell is acquiring specialized function. While acquiring specialized function, it remain inactive, or it has ceased its proliferation for temporary period. So it irreversibly lost its ability to proliferate. While acquiring specialized function, it has lost its ability to proliferate. So let's see the images now. See, interphase is a resting phase, very active phase during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. So it is subdivided into three phases: G1 phase, S phase, G2 phase. G1 phase is a period between M phase and DNA replication. S phase is nothing but where the DNA synthesis is occurring. Then G2 phase, it is a gap between DNA replication and preceding initiation of M phase. There is another G0 phase where the cell do not divide. They enter into G0 state. It is a resting cell. Almost all of our cell in our body is in resting phase for a it can be in resting state for a week, for a month, for a year. Okay, there are three stages or states: reversible G0 state, which is known as quiescent state; irreversible G0 state, that is senescent state; and terminally differentiated state. Reversible G0 state is where the cell remains metabolically active but no longer proliferate. Depending on the requirement of the organism, they resume the cell division. Okay, irreversible G0 state, which is a senescent state, where the dysfunctional cells have ceased completely the proliferation and they are permanently withdrawn. They have lost their proliferative potential. Terminal differentiation state, it is a course where cell is acquiring specialized function. At that point of time, the cell has irreversibly lost its ability. Next, we'll discuss about the time duration of each phase. That is time duration. 95% of cell cycle is spent in interphase. So interphase is the largest phase. Okay, G1 phase is 11 hours, S phase is 8 hours, G2 phase is 4 hours, constituting 23 hours. So 23 hours, in 23 hours interphase process will be happening. Then in M phase, 1 hour. It is a shortest phase where division happens. So mitosis, nuclear division. So M phase starts by nuclear division and ends by cytoplasmic division. So in our next class, we'll be learning about cell cycle control system. So which are all the aspects or elements helping the cell cycle control. So is there any component which guides or which supports or which regulates the cell cycle? Is there any component which stops the cell cycle or seizes the cell cycle and seizes the proceeding or the proliferation of cell cycle? So all these aspects will be dealt in the next class. Thank you.